point mutations or some other defect or derangement or alteration of immune or biological function, physiological function. But in terms of clear drug-associated events, such as the development of candidiasis in the case of 17 blockade, the equations describing or the formulas that fit the expected response will, both in terms of benefit and in terms of risk, side effects, will fall on these lines that are described mathematically as the Hill-Langmuir equation. They will be offset a little bit, so we need a higher level of response, a higher level of exposure in order to achieve the adverse effect compared to the benefit. That's what we call we call that the therapeutic window. And it will be somewhat parallel. They won't necessarily be exactly the same, but they'll be fairly, I would expect, fairly similar between the response curves and the side effect curves. They're just displaced by maybe an order of magnitude in terms of the exposure that's required.
Is it also something that you would expect with sonelokimab, as you've seen also higher rates of candidiasis?
Sorry, which one?
The Merck's nanobody. Is it something you also see?
The Merck's, so there's Merck AG, so the sonelokimab, or
yes.
Yeah. So the sonelokimab, we have yet to see what the actual potency or the binding characteristics of that nanobody might be. Once those are made available publicly, then one will be able to predict with, I think, a high degree of certainty what the relative benefit and risks of developing candidiasis might be. So it was already hinted to in our earlier phase study. The results indicate that we did see, at least in the upper dose arms, fairly high frequent occurrence of oral candidiasis. And that just reflects the fact that those were also the doses that resulted in the higher levels of effectiveness. So that we know that the same relationship will hold. I don't expect it to be any different from bimekizumab, whether or not the fact that it's a nanobody compared to a monoclonal antibody will make a difference. I suspect there will be subtle clinical differences, but I don't think either the particular overall benefit or the particular overall global side effect profile would be that much different.
Yeah. Regarding this sonelokimab and a nanobody, do you know when the phase 3 is due to start?
Sorry, I didn't get the last statement.
You're
actually quite quiet on the voice.
Sorry, I can speak
a very gentle voice.
Okay. Do you know when is the phase 3 due to start?
No, I don't think anybody does.
Okay.
So, yeah, I am not speaking out of turn, but Merck did not actually sought an external partner to do the phase 2 program. That partner is not in a position to carry out commercialization of the product, or they could yet be, I think, brought on board to look at a phase 3 program, but they're not really in a position to develop the drug. And typically, for a successful phase 3 program, you want the ultimate sponsor to be responsible for the program.
Yeah. Right, makes sense. And you mentioned that there could be subtle differences between bimekizumab and sonelokimab. In the presentation at the EADV, you mentioned that the nanobody could bring a more rapid response. Is that the subtle difference that you were talking about?
So that is just speculation. So at this point, it's very challenging to see what the differences might be. There's no question that being a smaller molecule, it's likely to have, generally speaking, likely to have better absorption characteristics. So instead of the rule of thumb for most monoclonal antibodies is about 60% bioavailability. Some are extraordinary, so we have 90% for risankizumab, but that's an outlier. Most of them sit pretty tight to the 40 to 60% band. Then we have a nanobody where potentially we could have higher absorption than that. We might get up to, say, the band I expect, given the much lower molecular weight, is likely to be about 10% better, maybe 15% better.
Okay.
And then there's access to the target. So being a smaller molecule, it will diffuse more efficiently. Having albumin essentially as a carrier, there may actually be conditions where we get active transport or semi-active transport, sort of enhanced transport to a target as opposed to just passive diffusion, which is typically the case for a monoclonal antibody.
Okay.
And even if one looks at diffusion, the diffusion of a small molecule is always going to be faster, greater than the diffusion that occurs, or the rate of diffusion that occurs with a large molecule. There are some little caveats because it depends upon the electrical charge and those other kinds of wonderful things. But the overall molecular or electrical profile of the nanobody is not going to be that exciting, nor is it that the case for a monoclonal antibody. So for the most part, it's really just the size that determines the diffusion characteristics. Right.
Okay. And there was actually another thing in your presentation. So looking at the efficacy, 50% of the patients achieved PASI 100 or free of disease. The verbatim free of disease really echoes a bit the IL-23p19